Your assignment for this week is to read the textbook, chapter 18, Comparing Two Means. And this is a really nice little chapter. It starts off just talking about examples, a lot of examples. And this is going to help you to build your intuition about what this test means and how you use it. It gets into some specific data sets. It walks through them step by step, does the calculations, and then gives you more examples. And that's exactly what we're going to do today in the lecture. So please do read the chapter. It'll help you a lot to understand what's going on in class. And it's, you know, kind of required. Oh yeah, and then there's a homework assignment. And the homework assignment, you will have all the tools to do it by the end of this week. And you have 2 weeks to do it. You're going to go out and get your own data, run a 2-sample t-test, and write a little report about that. And the third assignment I sent out was about the lab on Thursday. I sent you 2 .rmd files. Those are our notebook files. Up until now, we've been using the .r file extension. We've been writing R scripts and running our code in those. But from here on out, this week, we're going to start using our notebook. And those are a .rmd extension. And those will require you to upload some new packages. So I want you to all be sure that you can open them and that when they prompt you, there'll be a little line at the top in yellow saying install these packages, install those packages, make sure it works. Do that today. And if you can, or do it as soon as possible if your power's on. And if you have any problems, let us know as soon as possible so we can meet with you and try to work through any glitches, okay? We're going to, again, talk about the t-test 2-sample comparing two means, read chapter 18. So our learning objectives for today are to learn how to compare 2 means using a 2-sample t-test. By the end of this lecture, you'll know everything you need to know. Okay, by the end of this week, you'll be able to do it. We're going to go over the assumptions, the hypotheses, the calculations, and how to write conclusions, as well as how to differentiate among the different types of t-tests. There are 3 different types of t-tests. The first one we went over last week, the 1-sample t-test, and that determines whether the mean of a single sample differs significantly from a known or hypothesized population mean. And we use that when we have one group and we want to compare its mean to a known or hypothesized value. For example, our class height was the sample and the known or hypothesized value of the national average we compared. And we found out we weren't different, right? So that's neat. Then there's the 2-sample t-test with independent samples. And this compares the means of 2 independent groups to determine if they are significantly different from each other. And you use this when you have 2 groups and you want to compare their means. The third type of t-test is the paired t-test. And this determines whether the means of 2 paired samples differ significantly from each other. And that's used when you have 2 related groups and want to compare their means. For example, before and after measurements. There are 2 kinds of 2-sample t-tests, right? There's the 1-sample t-test and 2 kinds of 2-sample t-tests. The first one we use for independent samples. The individuals in both samples are chosen separately or they're chosen independently. For an example, compare the GPA of student athletes versus non-athletes. And the second kind, paired samples. The individuals in both samples are related or paired in some way because the same subjects are measured twice or they're matched pairs. And usually this is done to control for bias, like doing your study on sibling pairs. An example of this would be to compare the jumping height and men and women. If you compare the jumping height of men and women, but men tend to be taller, it wouldn't really be a fair comparison, would it? So what if you compared the jumping height of men and women, but you made sure that the men and the women that you're comparing for each particular jump are the same height? So you can see if there's a difference in jumping height based on sex and not necessarily based on some other factor like height, which would affect jumping. So here are some questions that you might want to answer with t-tests, 2-sample t-tests. Are there gender-based behavior differences in young wild chimpanzees? So if you randomly select 12 young chimpanzees and you tag them remotely with a dart and monitor their behavior, you can record the amount of time each young chimpanzee spends in contact with its mother. Then compare the amount of time spent in contact with the mother by the young males, as well as by the young female chimpanzees. That would be an example of a question you could potentially answer with a 2-sample t-test. And here, as I go through these examples, I want you to start looking for a couple of things, and I'm going to bring it up on the screen each time to remind you. What's the population of interest? What's the RV? That stands for response variable. We talked about this before. The response variable is the dependent variable or the Y variable. It's the thing that is potentially being affected by the EV, the explanatory variable. A good hint when you're looking for which is the response variable is there'll be units associated with it, right? The response variable usually has some units associated with it. It's the thing you measure. And in this case, the response variable is the time with the mother, and the units are minutes. The explanatory variable is sex, and there are 2 levels, male and female. With a t-test, we always have a numeric response variable and a categorical explanatory variable, and it always has 2 levels. Another question you could look at would be, does regular physical therapy help with lower back pain? Right? So how would you answer that? For example, you could say you take a randomized experiment assigned patients that all have lower back pain, and you put them into 2 groups. 142 receive an examination and advice from a physical therapist. Another 144 receive regular physical therapy for up to 5 weeks. After a year, the change in their level of disability from 0% to 100% is assessed by a doctor who doesn't know which treatment the patients had received. So what's the population of interest here? Patients with lower back pain, right? And what's the response variable? Change in level of disability. And the units here we have percent, right? It's a proportion. And what's the explanatory variable? Physical therapy, and the 2 levels are yes and no. They either got the therapy or they didn't. The 2-sample t-test hypotheses are pretty simple, but there are a couple of variations to pay attention to. The null hypothesis is really straightforward. You can state it in a few different ways. Okay, so the null hypothesis is really simple, but can be stated in a few different ways that I've listed here. These are just 3 different ways of saying the same thing. One is to say there is no difference in the population means. Another way of saying it is the difference in population means is 0. And an even more compact and simple mathematical way of saying it is that the population mean for sample 1 minus the population mean for sample 2 is equal to 0. The alternate hypothesis, or H sub A, is a little bit more nuanced depending on what you think might be happening in the population. The most simple way of putting it is to say there is a difference in the population means. Another way of saying that same alternate hypothesis is the difference in population means is not 0. right? It's the opposite of the null hypothesis. Or if you want to say it mathematically, for a one-tailed test, that's where you don't really know whether you think the difference between the population, one population and another will be greater or smaller. That's called a one-tailed test. Nope, sorry, I messed that up. Sorry, I just realized. This is the two-tailed. I was like writing this down. I'm like, wait, that's the two-tailed test. This is the one-tailed test. Let me fix that. Like, wait a minute. What am I doing here? I always find an error whenever I do this. Slideshow from current slide. Okay, great. Now I fixed this for you. I'll upload it again on Lalima for you. So the two-tailed test is you're saying we don't know if the samples are vary in one direction or another. We just think they're not equal. That's the two-tailed test. The one-tailed test says we think the population 1 mean is greater than the population 2 mean, or the population 1 mean is less than population 2 mean. So the two-tailed test has just the one possibility. The one-tailed test has two possibilities. Assumptions of the two-sample t-test. Very similar to the one sample. The first one is that we assume the data are a simple, random, or unbiased sample from the population. Again, you need to define your population of interest, and to be a really random, unbiased sample, every individual within that population should have an equal chance of being selected, right? And if they're not equally likely to be selected, you have to be very careful to see if there might be some systematic bias that would affect your results. The second assumption is that the response data are numeric or quantitative. And the third is that the observations are independent. That is, measuring one person doesn't affect your measurement of another person. Measuring one chimpanzee doesn't affect your measurement of another chimpanzee. The measurement of one student's height does not depend on or affect the measurement of the height of any other student, from our class example last week. And that the population is normally distributed. But lucky for us, t-tests are very robust to deviations from normality. So don't worry about it too much. We will look at it, we will check it, but the central limit theorem helps us out a lot, and we don't have to be too concerned if the data are not quite normal. Robustness, to go into this a little bit more, the two-sample statistic is most robust, most sort of, yeah, it's another way of saying robust is it's least affected by or least biased by, or is still very accurate, even when the assumptions are not perfectly met, as long as, one, both sample sizes are equal. If you have 2 samples from one population and 100 samples from another population, that can really throw off your estimates. Both sample distributions are similar. If one is very left skewed and the other is very right skewed, that might not be a good situation. You might want to try a different test. If your sample size is greater than 15, it is a simple random sample from the population, is close to normal, and has few to no outliers. You're probably in good shape. Or if your sample size is larger, if it's greater than 40, and it's a simple random sample from the population, even if the data are skewed or there are a few outliers, you're probably okay. Avoid the pooled two-sample T procedures. What? But I like swimming pools. Yes, but not this one. Not this one. So there are two versions of the two-sample T procedures, and they have very slightly different formulas and degrees of freedom. And this is kind of getting you down into the weeds here more than maybe you even really need to know, but I just want to give you a heads up at this stage, and later on, if it comes up, you'll be like, oh yeah, I kind of remember hearing about that, right? So we're not going to do the pooled t-test. The default in R is the unpooled t-test. So the pooled, what that means is that it assumes equal variances of the populations, and unpooled means it does not assume equal variance for the populations. If you look at this graph here, remember the histograms that we made before. When the variance of the population was small, the base of that bell was really narrow, like that blue curve on the left. And when the variance was really large, the base of the curve was really wide, like the blue curve on the right. Now, those are two normally distributed populations, but they have really unequal variances. Again, no problem. The t-test can handle that as long as you don't assume pooled variances. And that's usually just something that you specify when you're doing the analysis. And the reason is because the assumption of equal variance is kind of a hard, tricky thing to test. It's beyond our level. And also the test is just less accurate according to my reading. So we use what's called the Welch test for unequal variance, and that's the default test in R. So if you do nothing, that's what's going to happen. You're going to see the results say Welch test, and you'll say, okay, everything's fine. So here's another example of the two sample t-test. Now, when we're doing a t-test or any kind of statistical analysis, remember, we're not necessarily starting with a hypothesis. We're starting with a general idea about something that we think is going on in the world. Maybe it's from an observation you've made or an assumption that you have about the way things might be. Like you might see a picture like this. You see a mother smoking and she's got her daughter there and they look very cozy together. What's wrong with that? You know, maybe that's perfectly okay. But you might think, well, I don't know. I mean, maybe smoking harms children in some way. Maybe it has a negative impact on children. And well, maybe it does, but how would you quantify that? Right? What would you measure? So again, you might start with an idea of something you see going on in the world and you have to figure out how to quantify it. You need a response variable, something that you can actually measure, right? So here's a way that we're going to walk through of how to do that. So if you phrase your question more specifically, it helps you to kind of zero in on the right data to collect. Does smoking damage the lungs of children exposed to parental smoking? Okay, so now we're being more specific about the kind of damage, about the kind of harm. It's, oh, it damages the lungs, right? That makes sense. So how would you measure that? Well, one way of measuring it is the forced vital capacity, or FVC. It's the volume in millimeters of air that an individual can exhale in 6 seconds. And this is a measurement of lung health. So now, okay, we're quantifying the state of the lungs. If you measure the forced vital capacity for a sample of children not exposed to parental smoking and for a sample of children exposed to parental smoking, you can then actually do this experiment, and you can, not experiment, sorry, observational study, because you're not going to purposely expose children to smoke if not necessary. And so if you look at this little study example, what do you think is the response variable, right? Well, it's forced vital capacity, right? And the units are milliliters. And what's the explanatory variable? Whether or not the parents smoke. Parental smoking, yes, no. And what's your population of interest? Your population of interest is children, right? You're not measuring adults. Okay. So the population is children. Now you might have to then define, well, who's a child? Under 18, or are we looking at a specific age group, or is there another demographic that we want to consider? Again, your response variable is forced vital capacity, FVC in milliliters. And the explanatory variable is parental smoking with 2 levels, yes, no. Right? And so we're going to go more into this example. So the question again, is the mean FVC lower in the population of children exposed to parental smoking? I'm just restating it here because now we're going to talk about the hypotheses. So the null hypothesis for that would be the mean of the population of children with parents who smoke minus the mean of children who don't smoke. Sorry, the mean of the children with parents who don't smoke is 0, right? You take those two sample means, you subtract one from the other, and your difference is 0. There's no difference. So when you think about the null hypothesis, remember this is 0 here, 0 here. No difference. It's a 0. The alternate hypothesis is that, now this is, remember, the one-sided t-test, because we're actually looking to see, is the FVC lower in the population of children exposed to parental smoking? We think we know which way it's going to go. So our alternate hypothesis is actually that the mean FVC for children with parents who smoke is less than the mean for children with parents who don't smoke. So that's a one-sided t-test. And here are the results. You can put these in a little results table. I want you to pay attention to these results table because I want you to do the same thing with your own data. You'll have a heading here that's your explanatory variable, your 2 levels, in this case, yes, no. You put your mean, your standard deviation, and your n here, and you'll make a little table like this. That's part of your homework. And I'll show you a few of these examples. So in this case, the FVC for children with parents who smoke was 75.5, and for children with parents who don't smoke was 88.2. So it looks like it is lower, but is it a significant difference? Here's the maths. I mean, don't bug out on this. Like if you're like me and you see an equation and your eyes just glaze over, you're like, whatever, I'm going to completely ignore this. You can, but you know what? I think you could also do this. If you just hang on a minute and look, I'll walk through it with you and check this out. We're just looking at this T, okay? We're trying to figure out the test statistic, right? And we can figure out what each one of these things are. It's basic math. And then the test statistic is equal to the mean of the kids with parents that smoke, right? Right up here, 75.5, minus the mean for the kids of parents that don't smoke, 88.2, right? You plug that in here, right? Divided by, and what this big thing here is on the bottom is just the standard error of the difference between the means. It's also pretty simple. Look, it's just the standard deviation squared for the smoking group, which is variance, right? We already learned how to calculate that. We did it by hand, divided by the number of people in the smoke group. And then you add that to the variance or the standard deviation squared of the no smoking group and divide that by the number of people in the no smoking group. So it would be over here, you see 9.3 squared. That's our yes standard deviation, divided by 30. That's the n for the yes. 15.1 squared. That's the standard deviation for the no group, divided by the n, right? And then you just take the square root of that whole monstrosity. And then you get negative 12.7 divided by 3.24. That's a fraction with decimals. That's like 5th grade, right? We know this. And that's approximately equal to negative 3.92. And if your t statistic is large, it means your p-value is small, right? Big T, little p. So you also have to work out here your degrees of freedom. Okay, now this is a little bit of a monster too, but again, it's really not that hard if you just break it down step by step. So degrees of freedom, DF, equal to, again, we have the same kind of big chunk up here. We're just plugging in the standard deviation and squaring it for population 1, dividing that by the n, or the number of our observations for population 1. And then we add that to the standard deviation squared for population 2, divided by the observations of our sample 2. And then add those together, square those, divide that by 1 divided by the number of samples minus 1 times variance over number of samples squared, right? It's same thing. It's just the standard deviation and the number of samples, just keep plugging them in, plus 1 divided by the number of samples for sample 2 minus 1 times standard deviation squared for sample 2 divided by number of samples for or keep saying population, but this is a sample mean. Number of observations for sample 2 squared, plug that all in, plug in all those numbers, and you get about 48. And that's going to tell you your degrees of freedom. So there are many, many T distributions. There are many T distributions. Your book goes into it more. It shows you a bunch of different T distributions. Each T distribution has a slightly different shape, and you need to know that degrees of freedom so that you can get the right shape, so that you can put your T-stat in the right place to get your p-value. And back in the day, we used to use a book, but now we just plug it into the software, and we get here a p of 0.00014. So at an alpha of 0.05, we reject the null hypothesis. We reject the null hypothesis. The difference in means is not 0. The difference in means is not 0, right? There is a difference in means. So the way we want to write our conclusions here, I'm giving you this as a template, and I'll give you a few of these examples. This is exactly how I want you to write your conclusions for your homework and your reports. It's a sort of adapted form of the APA standard format. If you're in pre-med, you'll need to know this later, so might as well learn it now. So you could say lung capacity is impaired in children exposed to parental smoking compared with children not exposed to parental smoking. Now, we didn't actually say the FVC. We just went over to say, hey, here's the answer to our bigger question that we're trying to figure out. Is there harm done to children by their parents smoking? Yeah, it harms their lung capacity. It impairs their lung capacity. And we found that there was a pretty strong effect of that. Now I'm going to kind of show you this math simplified. And that is to say the T statistic, the test statistic, is equal to the difference between the sample means divided by the standard error of the difference between the sample means. And I wanted to resolve all this down to a simpler format so you could really get a feel for what's going on, that the test statistic is essentially equal to a proportion that's related to the magnitude of the difference between the means. And in other words, the magnitude of the effect of the treatment divided by some measure of the variability in that effect, right? The standard error of the difference between the sample means. We'll see this again and again in the parametric test. So I want you to have a kind of a grasp that this is a ratio of the amount of difference. So if the amount of difference between sample 1 and sample 2 was really small, and you had a large standard error, then what's going to happen to that test statistic? It's going to get small too, right? But if you have a really big difference between X1 and X2, then even if your standard error variation is fairly large, you'll still get a pretty big T. Breaking it down, the test statistic is essentially a ratio of the difference among groups to the differences within groups. And again, we'll see that more when we look at other tests. So keep this in mind right now. Okay, the confidence intervals. We need confidence intervals for reporting our results. People like confidence intervals on graphs and in your results section, and I'll require it, for your homework. The margin of error. We did this last week real quick, right? We looked at the margin of error. So the margin of error is your test statistic times that, remember this honking thing? We saw this before. This is just the standard error, right? The standard error of the difference between the means. So it's just the t times standard error. Same thing we did last week, actually. And that gives us our confidence interval. So here we would have negative 3.92 times 3.24 is equal to 12.7. And if you wanted to write your conclusions with confidence intervals, you could then say the difference in lung capacity, or FVC, for children exposed to parental smoking is 12.7 milliliters, plus or minus 12.7 milliliters here. T equals negative 3.92 and your P is 0.00014. Right? So you could do this math. It's doable. I hope everybody watched the video on confidence intervals. If you're confused about confidence intervals, go back and watch that video. It takes 5 or 6 minutes and it explains it really well. So here's another example of the two-sample t-test. And this is with seedlings, which I studied for my doctorate. I really love seedlings. I think they're super cool. And I'm also pointing this out to you because this might be something you can do for your projects, your independent projects, probably not for your homework because you won't have time. You only have 2 weeks, but you could do this for your class project. You could start by having a control and an experiment in which you put seedlings, and then to your control, you don't add fertilizer, and to your experimental pots, you do add fertilizer. And your question might be very simply, how does fertilizer affect seedling growth? How does it affect seedling growth? So the seedlings could be randomly assigned to pots with fertilizer or pots without fertilizer. Seedling growth in millimeters is measured after 4 weeks. The X variable, your explanatory variable, otherwise known as the independent or treatment or group variable, is fertilizer, right? And your 2 levels are fertilizer, yes, no. If you have more than 2 levels, you have to do a different test. The t-test is only for when you have 2 groups. Remember, 2-sample t-test. The data type for your X variable is categorical, right? It's a category, fertilizer, yes, no. We're not looking at the amount of fertilizer. We're just looking at whether or not you added it. Your Y variable, also known as the response or the dependent variable, is seedling growth in millimeters, not centimeters. That's a mistake. 4 weeks, it'd be hard. I mean, what species would grow fast enough that you'd get a response in centimeters? I guess it doesn't matter. It could be millimeters or centimeters, but you know, if you're measuring seedling growth, you better have something that grows pretty fast in 4 weeks to get centimeters. And the data type for your Y variable is numeric. So that meets the first bunch of assumptions, right? You've got randomly assigned treatments, and you've got a numeric Y variable and a categorical X variable. So fleshing out this example more, the question again, how does fertilizer affect seedling growth? The null hypothesis is there is no difference in growth between fertilized and unfertilized seedlings. In other words, the mean of the fertilizer group is equal to the mean of the no fertilizer group. Another way of saying that is that the mean of the fertilizer group minus the mean of the no fertilizer group is equal to 0. The alternate hypothesis is that there is a difference in growth between fertilized and unfertilized seedlings. And here you would simply say that the mean of the fertilizer group is not equal to the mean of the no fertilizer group, or if you subtracted the 2 means that they would not be equal to 0. Move this out of the way. That's as far as I can move it. There we go. Okay. So not equal to 0 for the alternate hypothesis. So are they not? What happens when we take some data and find out? So I made up some data. I made up some data. Actually, I kind of made up the data. I had the means and I had the standard deviation, but I didn't have the data. So I had R simulated for me, and I emailed you the code that I used to do this in the last email. I think I sent this yesterday. So if you just wanted to see how I go through this experiment and you wanted to run through it yourself, it should all just work. You don't actually have to upload any data. I'm just simulating the code within R. So the two different groups, again, treatment is fertilizer, control, no fertilizer, N equals 23. So by N equals 23, I mean 23 control pots and 23 experiment pots. And I kind of wanted to show you what this would look like if you were going to do it and you wanted to say your N equals 23. This is one N. This is one N. It has two pots, one control and one experiment. And I think I counted right. There should be 23 of these. And I'm bringing this up because this is something that people get confused about a lot. I see this mistake happen a lot. Even professional scientists sometimes get mixed up about what is an N. What is an N? And in this case, an N is a fully replicated trial of the experiment, which means it has to have the control and the treatment. Okay, again, going over the two sample t-test assumptions. So for seedlings, do we meet them? The data are a simple, random, unbiased sample. Yeah, assuming we just grabbed a bunch of seedlings, we didn't try to put the bigger one in the fertilizer pot and the smaller one in the non-fertilizer pot. We just randomized them and threw them all in the pots, and then we randomly assigned the pots all to a treatment, then that should be fine. The data are numeric. Yeah, check, right? We are measuring the height in numbers. So that's a quantitative variable. The observations of it are independent. Now, you could mess this up if you put the pots right next to each other. Maybe one is shading the other, right? So you want to make sure you actually set up your experiment in a way that one observation does not affect the other observation, and that the population is normally distributed. Now, this is not something we can know or necessarily create in our experiment. It's something that we have to examine with our data. But again, t-tests are very robust to deviations from normality. So don't worry about it too much. Don't let yourself get stressed out. Are my data normal? We'll check it. We'll try. We'll look at the rules for robustness. We'll do our best to get a big enough sample size, and probably it'll be okay. In this case, I generated normal data, so the data are normal. So the two sample t-tests, when I make a graph of the histogram. So this again counts the frequency of each value. So we had one seedling grown without fertilizer that reached this height, right? A height of 1. This is how, wait, height of, this is going to be 0 to 10, right? So we had one that was between 0 and 10 millimeters. Another, and we had 3 that were between 10 and 20 millimeters, right? 20, yeah, something like that, right? So you can count how many individuals fell into each size class. And when you look at the overall shape, you can see, okay, it's not exactly a perfectly symmetrical bell-shaped curve, but it's more or less. And then when you look down here at the fertilizer group, now that looks really darn good, right? That's a pretty nice bell-shaped curve right there. So I think we can proceed with confidence that our data meet all the assumptions of the test. So if we want to also look at the distribution, looking at the box plot, here's another way of doing this. And this also gives us an interesting idea of how to compare the spreads, as well as the starting to get an idea if the means might be different. We might also see that the medians are different. And we also see again that the data are pretty symmetrical and we don't have any strong outliers. And when you actually run the test in R, this is what the results are going to look like. All right. So again, at the beginning here, this is, I just copied and pasted this from R. This is what it's going to give you. So get used to looking at this. It's going to say a Welch 2-sample t-test. Remember, the Welch is the unpooled variance version of the test. Good. That's what we wanted. We don't want to assume equal variances, although looking here, we probably could, but why do that? Next, we want to get down to this line here that I just highlighted. We want to get to nitty gritty of the results, the t-stat 2.2427. That's a pretty big t-value with degrees of freedom 37 and p-value 0.03089. Now that p-value is less than 0.05. So assuming we set our alpha at 0.05, our p-value is less than that. So we reject the null hypothesis, right? And here the test gives us automatically the output of 95% confidence intervals for the difference between the means. So we look at the mean estimate of X and the mean of Y. So the mean of the fertilizer group and mean of the control group. We have 51.7 and 42.5. The difference between these two is bracketed by the 95% confidence interval. And I'll go into that more here. So here on this graph, I show a bar plot of the difference in means with a 95% confidence interval. So you can see here, this is the mean of the control group is the top of this bar. And these barbells here, those are the 95% confidence intervals. And here at the top of this bar is the mean for the fertilizer group. And this barbell here, this represents the 95% confidence intervals. And here's how we can write our conclusion. One way we can write our conclusion. Seedling growth increases with the addition of fertilizer by an average of 9.19 millimeters. So if we subtracted one mean from the other, it would be 9.19 millimeters, plus or minus 8.56 millimeters, 95% confidence interval. So if you go back to the results here, if you take that 9.19 millimeters and you subtract this number or this number, oh no, sorry, yeah, that's right. That's the number that you get. You get 8.56. So this is the confidence interval for the difference between the means, and you get it from calculating from the t-test. Again, I sent you all the code. You can do it yourself in R. And then you always want to include your supporting evidence by opening up the parentheses, writing your test statistic, comma, degrees of freedom, comma, p-value. You don't have to take the p-value to like 5 digits. 2 is fine. 3 is fine. Right here, this is good enough. Again, the R code is in the resources tab, so please do take a look at it. And briefly to review the P procedures, I'm putting this up again because I want to give you a mini quiz on this. And also next week, there will be a quiz on the different types of t-tests. And we're going to practice that today. So just to remind you, the one sample t-test determines whether the mean of a single sample differs significantly from a known or hypothesized population mean. So you're comparing a sample to a population, essentially. And you use it when you have one group and want to compare its mean to a known or hypothesized value. And the two sample t-test that we looked at here compares the means of two independent groups to determine if they are significantly different from each other. And you use it when you have two groups and you want to compare their means. And the paired t-test, the third kind of t-test, determines whether the means of two paired samples differ significantly. And this is used when you have two related groups and want to compare their means, for example, before and after measurements. And now we're going to look at this mini quiz. You're not graded on it, but do try to answer because this is going to help you on the actual quiz next week. So each situation requires inference about a mean or means. Identify each as involving either 1, the one sample t-test, 2, the paired t-test, or 3, the independent samples, 2 sample t-test. Okay, 1, acid rain, the acid rain example. You contact a random sample of 24 hydrological stations in California and obtain data on the acidity of rainwater collected at each station in 2000 and 2010. And of course, this is like 14 years ago. So you had to like call them and you couldn't just like get an API and download the data from the internet. Acidity is measured by pH on a scale of 0 to 14, right? The pH of distilled water is 7. It's always nice to include some irrelevant information in your word problems just to make sure you're paying attention. You want to estimate the average change in acidity of rainwater in California between the year 2000 and the year 2010. So you want to estimate the changes in these two samples. So do you think this is a one sample t-test, a paired t-test, or independent sample two sample t-test? What do we think? What do we think? Anybody want to guess?
I'm going to guess independent.
You think independent? Anybody else have another guess? Everyone agree? Disagree? What do we think? Okay, so in this case, actually, I think this is a paired t-test. Now, it can be a bit tricky, but you're repeating measurements on the same stations, right? This is like before and after. The year 2000 is your before, and the year after is 2010, right? And if you think about it this way, when you're thinking about if it's paired or not paired, or if they're independent or not independent, think about the variability. The variability within one station, one of those 24 stations, is probably less than the variability among all the different stations, right? Each station is measuring the pH of the rainwater at a particular place, and the rainwater at that particular place probably has a particular value that might change over time, but maybe is unique to that location as well, right? So the sample that you took in 2010 and 2014, 2000 and 2010 are essentially paired samples. All right, does that make sense? It's a tricky thing. That's why I'm like really kind of driving it home here. All right. Agricultural pests. I know we got some ag majors here, so I wanted to make sure I put this in here. Okay, agricultural pests. Agricultural pests can be controlled to some extent, either by using pesticides to kill them or by introducing a large number of sterilized males to diminish the species' reproductive potential. 10 large cornfields are treated with pesticides and 10 have sterilized males introduced. Corn yield at harvest time is then compared for the two treatments. Now, do we think this is a one sample t-test, paired t-test, or independent samples two sample t-test? Anybody else want to hazard a guess? One way to think about it is, how many total cornfields do we have? How many total cornfields do we have? Right here with the paired t-test, 24 hydrological stations measured twice each. We only have 24. If we had measured 48 of them, they would be unrelated, right? 24 this year, 24 that year, they're unrelated. In this case, we have 10 cornfields. We added pesticide, 10 cornfields. We did the sterilized male treatments. Assuming they're not right next to each other, could be a different experimental design, this would be an independent samples two sample t-test. Right? Next one, chemical analysis. So to check a new analytical method, a chemist obtains a reference specimen of known concentration from the National Institute of Standards and Technology. She then makes 20 measurements of the concentration of this specimen with the new method and checks for bias by comparing the mean result with the known concentration. Okay, what do we think? Is this the one sample t-test, paired t-test, or two sample independent test? Somebody's got to answer. One sample, yes, yes. You got it right. It's the one sample. And how do you know? Because you're looking at the reference specimen of known concentration from the National Institute of Standards and Technology, right? This is like the known concentration. This is your reference specimen. Like if you're comparing your blood pressure to the norm, the average blood pressure of people your age, or our heights to the national average heights, right? Good. So we got that. We got a lot more practice with that. We did that last week. Number 4, chemical analysis again. Okay, so another chemist heard about your way of doing things and thought, oh, that's interesting. Maybe let's try that. And he wants to check that new method. He has no reference specimen, but a familiar analytical method is available. He wants to know if the new and old methods agree. In other words, is there a difference in those meanings? He takes a specimen of unknown concentration and he measures the concentration 10 times with the new method and 10 times with the old method and compares those means. What kind of a t-test is that? One sample t-test, paired t-test, or independent samples two sample t-test? Somebody's got to answer. Ian, you already did twice. Somebody else. You have a 30% chance of being right if you guess anything.
Paired because it's the same specimen.
Tricky, huh? That one. Tricky. Yeah, I kind of thought that at first too. I was like, oh, it's the same specimen. But then I thought, well, wait a minute. How many actual samples did he take here? He did 10 and 10. He did 20. And like the measurement of that sample using one technique would not necessarily be affected by the measurement of that same specimen using the other technique. So in a sense, what we have here is like that specimen is our population, right? That's our population. And he's taking samples from that population to estimate the population mean. If he had a sample, right? And he took a sample from that population and he split it in two and he measured with the old method and the new method, that would be more like a paired t-test. But in this case, he just took a sample, a sample, a sample, a sample. So I think this is going to be an independent sample two sample t-test. You see how it's a little bit tricky? You have to think about it. It helps if you think about, well, what is the population of interest? What is the population of interest? How are these things connected to each other, right? And then you can even think of, okay, what's our response variable here? Our response variable here is, it doesn't even say actually, these don't really help us. In the cornfield example, yield is our response variable, and pesticide versus sterilized males, that's the explanatory variable. And yeah, each experiment's a little different. Okay, so our learning objectives, were they achieved? Here's our learning objectives. Did we learn how to compare two means using two sample t-tests? Well, we got a pretty good start. We went over the assumptions, the hypotheses, the calculations, and how to write the conclusions, and how to differentiate among the tests. On Thursday, we're going to go over this more. We're going to do some examples on our own, taking our own data. Now, if you all read my lengthy email, maybe you did, maybe you didn't because the power was out, but we're going to do data on ourselves. Please come to class. This is a fun class. It's going to be a fun class. Typically, everybody's like up and laughing. It's a really good time because we're going to take our pulse. Then we're going to do jumping jacks, 10 jumping jacks, right? And then we're going to take our pulse again and we're all going to enter it in the spreadsheet. We're going to see how it changes. And that's going to be a paired t-test because we're pairing each individual before and after. So come to class. If not, you're going to have to take your own pulse, do your jumping jacks on your own. It's just not as much fun. And then the other thing I want to bring up to you is that right now we have like a couple minutes left of class. Please go ahead and open those our notebooks right now. Just do it right now. Open them up, please. While I'm here, while Michelle's here, if anything goes wrong, we can help you. Just open them up. You'll see that they're going to say something like install this thing. I'll open mine too. See if I can get mine to open. Two-way t-test comparing means. I've already done it so that I don't think I'll have problems with it. But if you just installed R and you haven't opened a notebook before, it's going to look a little different than what we had before. It's going to have this thing at the top. It's going to have this thing at the top, these 3 dashes. And then it's going to have the title, the output, and it's going to have an output to an HTML notebook. Neat, huh? And then it's going to have these comments are going to turn up in blue, but they're not comments anymore. Now these hashtags are defining headers, changing the font size of our output. And here, where you see that little arrow, that's a place to prompt you to enter something. That's where you need to type an answer. And it shows up in green, and it also shows up differently in your HTML. So if I preview this as an HTML, it looks like this, right? You see this.
Can I ask for some clarification? This code was sent in an email, right? Or is it in Lalima?
No, I send it on an email because if I send it in Lalima, it comes out gobbledygook.
Okay, and the date when you sent it was today?
Just today, like an hour ago, a couple hours ago.
I just opened up my R Studio, so I'm going to show the class because it does prompt me to install. I'm going to move you.
Okay,
ahead.
good. Yeah, can you walk everybody through that? Actually, can you share your screen?
Sarah, I just wanted to say I was able to download the things you sent, but when I click on them, they don't open.
If you just click on them and they don't open, try opening up RStudio and then browsing to them and opening them in RStudio. And Brianna, I notice you end up with a lot of issues. Is there any way you can get to class in person?
Yeah, I was definitely gonna go this Thursday, so I'll just start going.
Yeah, there's something about your computer. I don't know, but I think that if you can get here, that'll help a lot.
Okay.
All right.
Yeah, that sounds good.
Okay, yeah, so here is R Studio. So see this little line right here where it's prompting me? It's saying packages are marked down in Google Sheet 4 required, but are not installed. So I'm just gonna simply click install. And you'll see it starting to do the installation right here.
So that one of them.
I will not come back up, unfortunately. But it prompted right here as soon as I opened up R. Have you opened up yours yet? Okay, did it not have that same little prompt?
If it didn't have the prompt, it might mean that those packages are already installed. Basically, when you open it up, it checks to see if those packages are installed. And if not, it says, hey, click here and install them. And it's so much easier. It just saves you that step.
Okay, cool. And then I can check it by trying to knit to PDF.
Maybe just try doing the preview because knit to PDF, you also have to install another package. If you go back to where it says knit,
Yeah.
and you just hit preview notebook, yeah.
Yep.
There
It popped up. Okay, yeah. That's perfect.
you go. Does anybody having problems with that? Any, Good. That really helps a lot, because if we don't run into glitches, we'll be able to just get through this lab so much more easily, and we'll have more fun.
I had something happen that says I need to install LaTeX.
If you try to knit to PDF, you will have to install LaTeX.
Oh yeah.
Did it give
But
you
instead
that
I
when you tried to knit, or before?
when I tried to knit to PDF, and I
Yeah.
guess I missed. We're knitting to what now?
HTML. Try knit to HTML.
Oh, okay. Can't get Google credentials.
I'm not sure the knitting is going to work, because to knit, you normally have to have all the steps of the code executed. Let me see but if you do preview, it should work. See if mine actually will knit. Yeah, it won't knit because we haven't loaded the data into it. I left that step blank. The nice thing about the notebooks is that they have a bunch of handy features built in, like prompting you to install packages. And also they allow you to write and run your code and export your results and your graphs and everything kind of in one step. And the way that it's typically done now, the best practices of science, is that you write everything in the script and you export it as a PDF, and that's your manuscript that you submit for publication. Like the whole report writing process is done in there, which creates a higher level of transparency of what people are doing, right? I could see all the manipulations that you've done with your data if I look at the underlying code. It's all in there. Sir, Zach.
Wait,